Genetic variation. We have studied how prokaryotes can help each other improve their chances of survival by making different proteins. But if asexual reproduction and binary fission creates copies of the original DNA, how do we end up with bacteria that can make different proteins in the first place? How does diversity arise in the bacterial world? Mutation and genetic recombination are sources of genetic variation for bacterial cells. Since bacteria can reproduce rapidly, new mutations quickly increase genetic diversity. More genetic diversity arises by recombination of DNA from two different bacterial cells. A major source of bacterial variation is mutation. Mutation is a type of change. Specifically, a mutation is a change in the DNA, the genetic code of an organism. A change in DNA sometimes leads to a change in an organism's characteristics. Although cells are very good at making accurate copies of themselves during division, they still make mistakes. During 1% of binary fissions, bacteria are mutated. So if 1 million E. coli cells divide, about 10,000 will have mutations. They will be new variations. Every generation has some mutants. Most mutations will cause the cell to die. Once in a while, however, a rare mutation will increase a cell's fitness. A mutation could allow a cell to attain antibiotic resistance. Maybe 1 in 1 million cells will continue to grow. If just a few bacteria carry mutations that allow them to resist an antibiotic, those few will survive, reproduce very quickly, and generate a new population of antibiotic-resistant bacteria. The model shows a random mutation giving rise to a resistant cell, shown in red. Within 3 generations, it had expanded in the population while other variants are stable, or yellow, or no longer present, pink. 3 processes bring prokaryotic DNA from different individuals together: transformation, conjugation, and transduction. These mechanisms of gene transfer and genetic recombination in prokaryotes lead to great diversity. 3 processes bring prokaryotic DNA from different individuals together: transformation, conjugation, and transduction. These mechanisms of gene transfer and genetic recombination in prokaryotes lead to great diversity. Transformation is the alteration of a prokaryotic cell's genes by the uptake of foreign DNA from the surrounding environment. For example, harmless Streptococcus pneumoniae bacteria can be transformed into pneumonia-causing cells. Click here to see an animation of transformation. The discovery of the genetic role of DNA began with research by Frederick Griffith in 1928. Griffith worked with 2 strains of a bacterium, a pathogenic S, or smooth strain, and a harmless R, or rough strain. When he mixed heat-killed remains of the pathogenic strain with living cells of the harmless strain, some living cells became pathogenic. He called this phenomenon transformation, now defined as a change in genotype and phenotype due to assimilation of foreign DNA. Here we have the rough strain, and the mouse lives. With the smooth strain, the mouse dies. With heat-killed smooth strain, the mouse lives, and with rough strain and heat-killed smooth strain, the mouse dies. Conjugation is the direct transfer of genetic material, plasmids, between prokaryotic cells that are temporarily joined. The transfer is one way. One cell donates DNA via a sex pilus, and the other receives the genes. The fluid which fills the cell is called the cytoplasm. Floating in the cytoplasm are the ribosomes and the bacterial chromosome, a double-stranded circular structure containing the prokaryote's DNA. Prokaryotes usually only have 1 chromosome, and the area where it is located is known as the nucleoid. The presence of an F plasmid gives the prokaryotic cell the ability to have fertility by forming a sex pilus. This allows the prokaryote to donate DNA to other prokaryotes in its colony, increasing their genetic variability. Note, fertility can also be present if the F factor is located in the bacterial chromosome. The presence of an F plasmid gives the prokaryotic cell the ability to have fertility by forming a sex pilus. This allows the prokaryote to donate DNA to other prokaryotes in its colony, increasing their genetic variability. Note, fertility can also be present if the F factor is located in the bacterial chromosome. The last mechanism that increases variability in the bacterial gene pool is transduction. Transduction is the process by which DNA is transferred from one prokaryote to another by a virus. Before we go any further, we need to explain what a virus is.